everyone, my name is Holly and I work in genealogy and local history at the Columbus Public Library. This video is part of a series about Ancestry 101, resources you can take home. In this series I will be introducing books that I've grouped together to help patrons focus on an area of genealogy interest. These are books that you can place on reserve and have delivered to any branch. So here is today's topic, DNA. If you've forgotten what you may have learned in school about how genes, chromosomes, and DNA are related, here's a quick review. The human body contains 100 trillion cells, and inside each cell is a nucleus, except for red blood cells. Inside each nucleus is where your 46 chromosomes are. You get 23 from each parent. Of these 23 pairs, 22 are called autosomes. Each pair is roughly the same length and have genes with the same arrangement. The autosomes determine everything except your sex, things like eye color and height. The chromosomes are filled with tightly coiled DNA, and genes are the segments of DNA that contain instructions to make proteins, which is the building blocks of life. Here is a lineup of one person's chromosomes by size, and they're numbered 1 through 22. The 23rd chromosome in this person has both an X and a Y, meaning the person is male. The stained chromosome photo shows the segments of DNA that are the genes, the dark bands on the chromosome. So what makes up DNA? DNA is a long molecule that is made up of repeating nucleotides. And nucleotides are made up of 3 parts. There's sugar, a phosphate, and a nitrogenous base. There are 4 types of nitrogenous bases, which are represented by the first letter in each word. A is for adenine, G is for guanine, C is for cytosine, and T is for thymine. Each base pairs up with only one of the other letters. A's always pair up with T's, and C's pair up with G's, like this. Here you can see how the bases pair up and are connected with a sugar and a phosphate to make a nucleotide. Here's a more colorful example of the base pair rule. And finally, the shape of DNA, the famous double helix. So why do we send in our saliva to get tested? To compare it with other people's DNA to see whether there is any genetic match somewhere, and if so, to determine how we share a common ancestor. Here is a simplified version of how the most common type of test works, the autosomal DNA. From your sample, DNA is extracted from your 22 chromosomes, the autosomes. Copies of your DNA are made. Then samples are taken at over 700,000 or more locations to see the nucleotide value at each location. Remember, the nucleotide has 3 parts, sugar, phosphate, and nitrogenous base. So there are 4 possibilities, adenine, thymine, cytosine, or guanine. Also remember that you have 2 parents, each contributing one of their chromosomes, so you will have 2 nitrogenous bases, one from your father, one from your mother, at that location. Without more information, it's not possible to tell which is paternal and which is the maternal chromosome. In this example, on chromosome 1, in a specific location and position, an A and a G are identified. And in another location and position on the same chromosome, a T and a C are identified. Your DNA will be compared to others who have taken the test at the same company. If you test at more than one company, your results may vary due to the difference in the reference population of test takers. Each company establishes minimum lengths of DNA segments to determine whether you are a genetic match to someone else in their database. DNA length is measured in units called centimorgans. It's abbreviated with a lowercase c and an uppercase m. According to the National Human Genome Research Institute, 1 centimorgan translates to approximately 1 million base pairs of DNA sequence in the human genome. Ideally, the testing companies want to identify only those individuals who share a common ancestor within the past 300 to 400 years, what may be called a genealogically relevant time frame, while also excluding individuals who share a common ancestor more than 500 years ago. As you go back in time, you lose half of each generation's DNA, so when you reach your fourth great-grandparent, there's only a 1.5% chance of inheriting any of their DNA. At the end of the presentation, I've included links to videos that I found to be helpful in trying to understand DNA. But now, here are 2 library books about DNA and genealogy that you can check out. Trace Your Roots with DNA by Megan Smoleniak and Ann Turner, and The Family Tree Guide to DNA Testing and Genetic Genealogy by Blaine T. Bettinger. The authors of Trace Your Roots with DNA are both renowned genealogists and experts in DNA. Although this book is somewhat out of date, the technical aspects of the tests are explained well, and the book is an easy read for those who are not familiar with either DNA or DNA testing. After a brief introduction to genealogy and genetics fundamentals, they explain the types of available testing, what kind of information the tests can provide, how to interpret the results, and how the tests work. A question authors of both books want readers to ask themselves before spending money on any test is, what is it you want to find out? For example, are you trying to find a surname connection? Is your paternity uncertain? Or are you simply putting your traditional genealogy research to the test? This makes a difference in which tests to take. The book includes case studies of historical mysteries, like how the remains of the Romanov family, an unknown Vietnam soldier, and a baby that died on the Titanic were eventually identified. There's also an interesting section on how the FBI uses only 13 markers to identify felons in the CODIS database. Our next book is also authored by a DNA expert, Blaine T. Bettinger, titled The Family Tree Guide to DNA Testing and Genetic Genealogy. This is his 2nd edition and was published last year. This is a book I purchased for myself after borrowing it from the library. For a subject like DNA that can easily get too technical, it's well written with lots of colorful graphics, and it gets a 4.32 out of 5-star rating on Goodreads. There are separate chapters on each of the 4 kinds of DNA tests currently available. Autosomal DNA, that tests the 22 of the 23 pairs of chromosomes, everything except the sex chromosome. Y chromosomal, that helps you track down your male line descendants. Mitochondrial DNA, to discover your female ancestors. And X chromosomal. There is an excellent guide to help you choose the right DNA test by answering the question, why are you testing? It takes you through a decision tree that will recommend 1 or more of 5 testing companies: Ancestry DNA, 23andMe, Family Tree DNA, MyHeritage, or Living DNA. The author covers what are called third-party autosomal DNA tools that you can explore once you receive your DNA results. Some of these are free or charge a nominal fee to access more in-depth analysis of your DNA. Companies like DNA Painter, GEDmatch, DNA GEDCOM, Genome Mate Pro, Promethease, and others. The book includes charts and worksheets like the most recent common ancestor, DNA cousin match worksheets, and ranges for centimorgan matches for your relatives. Here are links to 2 videos that might help you learn more about DNA. Thanks for watching and have a great day.